Hello, biochemistry students. Thank you for letting me know which were the muddiest points when we talked about gluconeogenesis. And I tallied the cards you handed in, and some of the most common questions were, where do we do gluconeogenesis and why? And how does it relate to glycolysis and some of the other cycles we've talked about so far? So in terms of where, gluconeogenesis occurs in the liver, primarily in mammals. Some gluconeogenesis also occurs in particular cells of the kidneys and in the epithelial, the intestine, but we're going to focus on the liver because that's where most of it happens. So this is when you're producing sugar, you're producing glucose from non-carbohydrate sources. So you're taking amino acids, for example, that make up the proteins of your skeletal muscle, and you're breaking down that muscle using those amino acids to feed into gluconeogenesis to go from pyruvate to glucose. That glucose will then be exported out of the liver in a regulated manner to other tissues so that they can use the glucose. Because when do you do gluconeogenesis? You do it when you do not have any glycogen stores left. So when you need glucose because you haven't eaten, you'll first break down your glycogen stores because that's the simplest way. Glycogen's a bunch of glucoses linked together in a polysaccharide. But if those aren't there, you still need to make glucose for your body. There are parts of your body that only use glucose or preferentially use glucose. And this is a survival mechanism that we've evolved to have. So when the glucose goes to the other tissues, glycolysis will occur there in those other tissues to make ATP to do all the cellular work those tissues and cells need to do. Now within the cells of the liver, glycolysis is going to be downregulated when your liver is working hard to make glucose for the rest of the body. That's what it means when the textbook says gluconeogenesis and glycolysis are reciprocally regulated. That means within cells that do both, primarily talking the liver, when it's time to do gluconeogenesis and make a bunch of glucose, the use of glucose will be downregulated. The use of glucose will be limited so that it gives a chance for those liver cells to make a bunch of glucose and let it leave the cell to go to other places in the body. Now, other cells in the body will accept the glucose and do glycolysis and make their much needed ATP. How gluconeogenesis is related to glycolysis is that gluconeogenesis is creating sugar for glycolysis to occur in extrahepatic tissues. Now the glycolysis will provide some ATP as pyruvate is made, and then the bulk of the ATP will be happening after pyruvate's fully oxidized, and the citric acid cycle and oxidative phosphorylation by the respiratory chain or the electron transport chain will get you much ATP. And so your body needs to make glucose because you're not eating and your glycogen stores have been depleted. So to summarize this concept of why you do which pathway, glycolysis happens when a cell needs ATP. And glycolysis can happen when glucose is available. Now, gluconeogenesis happens, and I should say for glycolysis, all eukaryotic cells. Prokaryotic cells do it too, but we're focusing on mammals right now. And with that focus, we're looking at gluconeogenesis happening primarily in liver cells. And this happens when your body needs ATP, your cells need ATP, but there's no glucose available for it. So glucose from diet or stored glucose in the form of glycogen is not available. I hope this clears up the why and the where. And I want to add another level of who, because this came up too, is, well, what are the precursors for the glucose made from gluconeogenesis? And you will use citric acid cycle intermediates and the amino acids from your proteins to serve as carbon skeletons to make your glucose. Remember, you have to end up with a 6-carbon sugar to get your glucose. Where are those carbons coming from? And so your amino acids will be converted into pyruvate, for example, to feed into gluconeogenesis. There were also a few questions about lactate and how lactate fits into things. So in lecture we talked about how lactate's produced for us in anaerobic muscle during lactic acid fermentation, and the lactate's going to travel from the muscle to the liver so that it can get inputted into gluconeogenesis. Now, this is going to happen when anaerobic muscle's working hard, but you're not eating, so you don't have any glucose coming in from your diet, and you've depleted your glycogen stores, so you don't have any glucose coming from there either. Now, the conversion of lactate to pyruvate produces an NADH, and that's very helpful because one of the later steps of gluconeogenesis requires NADH. So by having an early step of gluconeogenesis create this NADH, you're providing something that's necessary for a later step. What's that later step? Let's draw it out together. From PEP, you go to 2-phosphoglycerate, which goes to 3-phosphoglycerate, which goes to 1,3-BPG. So I'm just drawing gluconeogenesis now, which goes to G3P. And this step requires NADH. And this step is going to be happening in the cytosol. So this NADH is going to contribute to the pool of NADH in the cytosol and help this reaction happen. Now G3P, I'm just going to say etc. because we go all the way back to making glucose here. But I wanted to highlight that step which requires NADH. So that's great. NADH is in the cytosol for gluconeogenesis, when you start with lactate, but that's not the common path for gluconeogenesis. The reason it's not the common path is because you're not normally doing anaerobic respiration. You're normally doing aerobic respiration. And so usually the source of the carbon, or I should say the first step of gluconeogenesis, is going to involve pyruvate, not lactate. And so when this happens, we've evolved to do this multi-step situation at the beginning of gluconeogenesis. And what it does is that it allows for us to get NADH created in the cytosol early in gluconeogenesis, so that there's going to be some there when the later step occurs. And so that's why we evolved to have this multi-step or multi-reaction first bypass step. So I think the text refers to it as bypass reaction, but let's, for clarity's sake, call it a bypass step because it indeed involves 3 reactions. So our bypass step number 1 involves pyruvate being converted to oxaloacetate. Then oxaloacetate can't leave the mitochondria, but malate can. So it gets converted to malate. Then in the cytosol, malate gets converted back to oxaloacetate. And that step allows an NADH to be created in the cytosol. And that's going to help that later step of gluconeogenesis over here. So it's really cool in that it's like if you need to do something later, but you do something now to set you up for success later. That's essentially what we're looking at now. So that several students mentioned that they were confused about the why was all this movement happening. And this is primarily the reason why we've evolved to have this mechanism is this movement of intermediates from the mitochondria to the cytosol, where the rest of gluconeogenesis happens, allows for reducing power to be added to the cytosol in the form of NADH. So one student pointed out, are you telling me gluconeogenesis is 13 steps and glycolysis is 10 steps? And yeah, you could look at it that way, because bypass step 1 has 3 reactions to it. So gluconeogenesis, we could look at it as 13 reactions, and glycolysis has 10. And so bypass step 1 has 3 reactions in it, and we're looking at bypass step 1 here. So I think that covers a lot of the common questions that were asked on the index cards. And I just wanted to point out for some of these other figures, maybe do some extra annotations while we can. So here's bypass step 1. Now there's 3 reactions for that. This figure is not showing all 3 of the reactions, so there's the malate dehydrogenase step that's not being shown. This is bypass step 2. So, and this is bypass step 3. These are called bypass steps because these steps in glycolysis cannot just go backwards using the same enzyme. All the other steps do use the same enzyme and just go backwards. So that was why I made such a big point of it early on, to talk about how reactions, if they're thermodynamically reversible, that enzyme that catalyzes the forward reaction can just as well catalyze the reverse reaction. Really helpful is this table, which puts the sequential reactions of gluconeogenesis in, and I wanted to point out this is the place to see the summary equation for gluconeogenesis. And I wanted to give you my reasoning for splitting this figure in half during lecture, because I thought it was overwhelming to look at both the pathways at once. And also, the two pathways would not be happening at the same time necessarily. You'll probably be doing this most of the time because you're not producing lactate. And finally, I wanted to point out several people asked about, well, where does the pyruvate come from? What feeds into gluconeogenesis? And so amino acids from your muscle get broken down to create either pyruvate or citric acid cycle intermediates, which can form oxaloacetate to feed into gluconeogenesis. So that's why, for example, if you're trying to put on a lot of muscle mass while you're bodybuilding, it's crucial to get enough protein in your diet, because we don't really have a storage place in our bodies for amino acids. We store glucose as glycogen. We store fatty acids as triglycerides, but we don't store amino acids as anything. We do have amino acids linked together as proteins, but that's not a storage. Those aren't storage sites. Those are actual things like our muscle, our skeletal muscle, and they're doing things. So we don't want to deplete our skeletal muscle, but we will in a pinch. And so that's one of the reasons why people are very cognizant of their protein intake when they're trying to be fit, put on muscle mass, whatnot. I don't know. I've just heard about it. I just read about it in books. Hope this illuminated some questions, and if you do have more, please comment or talk to me in class. Thanks so much.